mute. Two people have entered the waiting room for this meeting. See waiting room. Unakuja hivi. Unakuja hapa. In waiting room.
Yes.
Hello, good morning.
Unamsikia?
Hello, good morning.
Good morning, Mr. Mungai. How are you?
I'm fine, thank you. What about you?
We are fine. Thank you. Now we can hear you well.
Sorry?
We can hear you very well. Now, madam can proceed.
All right, thank you. Thank you.
Thank you. Very much. On.
Okay. Come here.
Bilembu enda uko sitting. Kai, honey, come and show me waiting room.
I believe there is a meeting. So you can just continue. There is somebody who is admitting and you can continue. Just continue. There's somebody who is
Good morning, students. So just leave waiting room.
Say hi to them.
So students, I hope now we are going to start our lesson. Good morning. I'm Madam Sanaipe, going to take you through biology lesson today. And we are going to continue from where we stopped last time. Last time, we were discussing on the topic cells, and we discuss the microscope. The two types of microscopes, the light microscope, the electron microscope, and care how to handle the microscope, and also how to use. We had also discussed magnification, resolution, and we had also discussed a generalized animal cell and plant cell as seen under light and electron microscope. Now, today, we are going to look different components of a cell. Last time we discussed, we said that we have what we call component of a cell. In component of a cell, we have what we call inclusion, and we have what we call, inclusion. These are components are found in a cell. And we have what we call organelles. These are two components of a cell. We have what we call inclusion, and we have what we call organelle. So last time we stopped at inclusion and organelle. Now, today we are going to discuss the component of a cell. And the first component we are going to discuss here, inclusion. So what are inclusions? We said that inclusions are non-living components in a cell. These are non-living components in a cell. An example of this non-living component in a cell, we have starch, we have glycogens, we have amino acids, we have glucose, we have fats and oils. So let's go to organelles. What are organelles? What do you understand by the term organelles? When you are told define an organelle, how do you define this term? We define organelle as living components in a cell. These are living components in a cell. And without this organelle, no life in a cell. Students, do you remember when I was introducing this topic on the cell? What did I say? What is a cell? What did I say? What is a cell? I said that a cell is a basic functional unit of a living organism. I say that a cell is a basic functional unit of a living organism. Therefore, meaning if this cell is a basic unit of a living organism, it means that no life without a cell in any living organism. And student, do you notice a living organism? We have two types of living organism. We have plants and we have animals. Meaning that both plants and animal have cells. Both plants and animal have cells. Student, I hope you are summarizing and you are writing something. So, see this, we have types of living organisms. We have plants and we have animals. Meaning, for plants and animals to have life, it means that they should have cells. They should have cells. Cell is microscopic, very tiny. You cannot be able to see on your naked eye. And that's why when I introduce this topic on a cell, I mention a microscope, an instrument that helps you to see tiny microscopic organisms. Therefore, without a microscope, you cannot be able to see a cell because these are structures which are found in the body of plants and animals, and they are very, very, very small, very tiny. You cannot be able to see using your naked eye. Therefore, you have to use a microscope. Therefore, we say that a cell is a basic functional unit of a living organisms. So let's come back. Without organelle, there is no cell. The organelle are the one making a cell. And the cell are the one making the body of plants and animals. Therefore, when we say organelles, these are components found in a cell. And this component found in a cell, they are life. They are alive. Organelles are living. It is defined as living component of a cell. They are defined as living component of a cell. Good. I hope you have understood up to that point. Now, we are going now to start from mitochondria, which is one of the organelles found in a cell. Last time we had discussed the structure, what is enclosing the cell, and we said that this structure that is enclosing the cell is known as a cell membrane. And we say that a cell membrane, it is semi-permeable. It does not allow just anything to go through. Since it is because it is semi-permeable, it means that it is selectively allowing movement of substances in and out of the cell. So we don't just say it allows substances. These are structures that enclosing the cell. These are structures that is enclosing the cell. And also allowing substances in and out of the cell. And does not just allowing any substance, selectively allowing. So anytime you are told you give the function of a cell membrane is that structure surrounding a cell of a plant and an animal. Remember that a cell membrane is found in both plants and animals. It is found in both plants and animals. Therefore, its work is to enclose the cell content. It is enclosing organelle. It is enclosing this inclusion. And on the other hand, it also allows selectively movement of substances in and out of the cell. Now, students, that's just a reminder of what I taught last time. Now, today, we are going purposely to discuss on organelles. And the first organelle that we are going to discuss is the mitochondria. A mitochondria. How do you describe mitochondrion? It's known as mitochondria. Check the spelling. Check the spelling, because biology spelling mistake is not a worded mark. We have mitochondrion, that is singular, and we have mitochondria, plurals. Mitochondria, plurals. Kindly check the spelling. Most of the students do not know the spelling of this organelle, which is very simple. We can also say mitochondria. So mitochondrion, that is singular, and mitochondria, plurals. How do you describe the shape of this organelle? We describe the shape of this organelle as a sausage-like. Students, I hope everybody have ever taken a sausage. So it's a sausage-like or a banana-like. Therefore, we describe the shape as a sausage shape organelles. And if we give the description of this organelle is surrounded by two membranes. It is very easy to identify this organelle inside a cell. You see, now in a cell, you can able to see very many organelles. We have very many organelles. We have smooth endoplasmic reticulum, we have probe, we have lysosomes. We're going to discuss all of it. We have centrioles and many other. So how do you able to identify a mitochondria from that cell? It is a sausage shape. Apart from being a sausage shape, just say, let me see if I can able to see a sausage shape structure so that you rush on it. You say that's a mitochondria. No. It's a better way that you can able to identify a mitochondria inside a cell. Okay, mitochondria is usually surrounded by two membranes. We have the outer membrane and we have the inner membrane. But remember that this is not the only organelle with two, surrounded with two membranes. We also have another organelle known as the chloroplast. The chloroplast is also surrounded by two membranes. But student, how will you able to differentiate between the two? Okay, now I want just you to know that it's not only the mitochondria having two membranes, the chloroplast also have. We are going to discuss the chloroplast later, but now we are now on the mitochondria. But I want just to remind you that it's not only the mitochondria having two membranes, also have. But how will you able to differentiate the two? How will you able to differentiate the two? Okay, write this different. Mitochondria stroke. We make a table, kindly make a table. You write mitochondria, and then you write the chloroplast. How will you able to differentiate between the two? Because both have got two membranes. Okay, and the mitochondria, you can able to see that the inner membrane of the mitochondria, it is folded. It is highly folded. So that's the difference. But when you look at the chloroplast, the two membranes are smooth, the two membrane are smooth. They are smooth membrane. But when you look at the mitochondria, the outer membrane is smooth. The inner membrane is folded. The inner membrane is highly folded. That's the difference you should know. So students, let's continue the mitochondria. So we have already described the mitochondria as this an organelle with the two membrane. It has the outer membrane which is smooth. The inner membrane is highly folded to form structures known as crystal. Student, this question is always very fun in an exam because sometimes you are asked, mention the line is pointing one of the folded structure. When you look in your notes or in your textbook, you'll realize that the two plain lines are the one pointing the folded structure, hence the name crystal. C-R-I-S-T-A-E, crystal. So then what if it is one? What if it is one? This question was in your midterm exam. What if it is one? What if it is one? We refer to as crystal. I hope you are summarizing your notes because I'm going to ask these questions later. Therefore, we have the inner membrane is highly folded to form structures known as crystal in plurals. Singular if it is one structure, one projection. Remember, there are several projections. Those several projections, those folding, that's the one we refer to as crystal. If it is one fold, it's referred to as crystal. It's referred to crystal. So, this is enough descriptions of mitochondria. And these foldings are very, very important in these organelles. But later on, we are going to see the importance of this folding known as crystal. First of all, let's see other structures, or let's see other components of this mitochondria. We have said that the mitochondria consists of our outer membrane, consists of inner membrane that is folding to form structures known as crystal, and it also consists of a fluid media where chemical reaction take place known as matrix, M-A-T-R-I-X, known as matrix. This fluid medium is very important because it's the one increasing the surface area for the chemical reaction to take place to produce energy. Good. That's a description of mitochondria. Now let's go to the function of the mitochondria. Student, if you are told you give function of a mitochondria, how will you give the function? What is this organelle is responsible for energy production? But you don't just rush and start saying that it is energy production or it is for respiration. That's wrong. We have better terms we use in biology. And if you master these terms, you are going to be a very, very good student in biology. Just take notes on this. Therefore, when you are told you give the function of mitochondria, you give it a site for respiration. Site for respiration. We usually mark that term, site. We mark the term site. Therefore, when you are told you give the function of mitochondria, it's just a simple and short answer. You just say site for respiration. But if you end up writing for respiration, that's very wrong. Let me assure you, that's very wrong. So you just say site for respiration, full stop, and you're going to get your good mark. So this is another question which is mostly asked, which is very important, basing on mitochondria. Adaptation of mitochondria to its function. How is it adapted? For it to be able to carry out this function of energy production, how is it adapted? How is it adapted? Adaptation of mitochondria to its function. One of the adaptation is that the inner membrane is highly folded to form crystalline to increase surface area for attachment. The inner membrane are highly folded to form crystal to increase surface area for attachment of respiratory enzyme. Kindly note that for attachment of respiratory enzyme, without the word attachment for respiratory enzyme, I am assuring you that's very wrong. So kindly student, whenever you are told you give the function, you give the adaptation of mitochondria to its function, remember to say its function is the inner membrane is highly folded to form crystal to increase surface area for attachment of respiratory enzyme. Or you can even omit the word, the inner membrane is highly folded to form crystal. You can also say simpler, they have crystal to increase surface area for attachment of respiratory enzymes. Good. That's one of the adaptation. The second adaptation of mitochondria to its function is that they have a fluid medium known as matrix where enzymes reaction place. Have a fluid medium. They have a fluid medium known as the matrix where enzymes reactions take place. So those are the two adaptations of mitochondria. Kindly student, that question is very, very important in exam. It was even in last year KCSE. So kindly master the structure, the parts, the function, and the adaptation. I hope you have understood up to that point. So let's look some of the consideration of the mitochondria during this is a KCSE marking techniques. It's a biology KCSE marking technique. So one of the reminder when answering a question basing on mitochondria, the function of mitochondria is not respiration, but site for respiration. I think I have mentioned that before. The function of mitochondria is not respiration, but the correct technique in KCSE is site for respiration. another reminder we are reminded is that crystal increases surface area for attachment of enzyme. I think I've mentioned that before. The word attachment for enzyme should be mentioned. Crystal surface area for attachment of respiratory enzymes. That's all. Thank you. So now let's continue with the second organelle. The second organelle we are going to discuss is the endoplasmic reticulum. We have two types of endoplasmic reticulum. We have smooth endoplasmic reticulum and we have rough endoplasmic reticulum. Student, how will you able to differentiate between the two in a cell? Let's say, for example, you are asking an exam to identify a smooth or a rough endoplasmic reticulum in a cell. How will you be able to differentiate between the two? From the term rough, what does this mean? From the term rough surface, and we have what we call smooth surface. Like using jokes while teaching because it helps you to understand biology and enjoying it. Let's take an example of adolescence. When you are at your adolescence, some of the youth have what we call pimples in their face. That is rough face. When you are that age, you are now above that adolescence, you start having a smooth face. Now I can able to see, you are now understanding what I mean by saying rough and smooth. Take an example when you are at adolescence, you have a rough face, pimples all over in the face, meaning the surface is rough. But to reach a time, you start gaining a smooth face. So when you say rough endoplasmic reticulum, it means this organelle, it is rough. It has got some roughness, some particles on top of it, on its surface, sorry, not on top, on its surface. But when we say smooth endoplasmic reticulum, it means ni nyororo, smooth kabisa. So, first of all, we have to describe the structure of endoplasmic reticulum. We describe the structure as these are interconnected channels. It's like you have a branch. You can't cut a branch with different, with other branches. So you get that when we said endoplasmic reticulum, these are series of interconnected channels, a series of interconnected. But you see that when you say that series of interconnected channels, surface is smooth. That's referred to as a smooth endoplasmic reticulum. And we have this called rough. When you say that interconnected channels rough on the surface, this referred to as rough endoplasmic reticulum. So students, now let us start with rough endoplasmic reticulum. What is it causing it to be rough? What is it causing it to be rough? Okay, we have some organelles which are found on the surface of this endoplasmic reticulum, known as ribosomes. Ribosome from the ribo. This is a circle-like. When we say ribosomes, how do we describe the shape of ribosomes? These are spherical shapes. If you have ever seen a ball, all of you, I know you love football, so I hope you have ever seen a ball. So, and even when you are young, I hope you have having those small balls you play around with. So when we say ribosomes, these are balls-like structure. They are sphericals from the name as spherical in shape. These are circles-like structures which are found on the surface of endoplasmic reticulum. Hence the name rough endoplasmic reticulum. So student note here. Apart from these ribosomes found on the surface of this endoplasmic reticulum, they are also suspended in the cytoplasm. Sorry. Therefore, when we say rough endoplasmic reticulum, it means that they're the one found on the surface. They're found on the surface all over. They're found on the surface of this reticulum. Hence the name rough endoplasmic reticulum. Therefore, these others, you get that when there are no ribosomes on the surface of this reticulum, it's referred to as smooth endoplasmic reticulum. So we have rough, and what is causing to be rough? Ribosomes are found on the surface. And if they don't have this ribosome, it's referred to as smooth endoplasmic reticulum. So students, let's look at the function. Let's look at the function of the two types of reticulum. Note we have smooth and we have rough. We have smooth and we have rough. So let's look at the function of smooth. How do we give, or how do we describe their function? So we said that the function of smooth endoplasmic reticulum synthesize and transport lipids within a cell. Synthesize means that they manufacture lipids. Lipids. What are lipids? Students, what are lipids? Kindly note that lipids, these are fats. These are fats and oils. These are fats and oils. These are fats and oil. As the one we refer them as lipid, a general name. Let us say fats and oil, we give a general names known as lipids. So the function of smooth endoplasmic reticulum is to synthesize. It is synthesize and transport lipid within a cell. The word within a cell should be there. Without within the cell, your answer is wrong. You see, biology. Biology is very interesting. Then it's a very interesting topic. if you know the techniques of observing during answering in an exam. Very simple subject that you can get a free A, very free A. If you observe the techniques, the KCSE marking technique, and that's what when I'm teaching, I'm mentioning what you should consider. So when you are told you give the function smooth endoplasmic reticulum, you say that synthesize and transport lipid within a cell. You can get it in the textbook. It is written synthesize and transport lipid, full stop. But kindly remember to add the word within the cell. We also have RAF endoplasmic reticulum. The work of the RAF is to transport proteins within a cell. It does not synthesize. It's only playing the role of transporting. Remember, we have just said what is causing it to be RAF? Ribosomes. Ribosomes are the one making this organelle to be RAF, meaning ribosomes are the ones synthesizing the protein and being transported by RAF endoplasmic reticulum. So them being on the surface of RAF endoplasmic reticulum, they are very, very important. They help in synthesizing the protein. After synthesizing, RAF endoplasmic reticulum plays the role of transporting. Note that? But when we say smooth plays the role of synthesizing, but at the same time after they synthesize, they transport by themselves. But RAF only play one role of transporting. Now we have so far discussed 3 types of organelle: Mitochondrion, smooth endoplasmic reticulum, and RAF endoplasmic reticulum. Good. Now let's go to another organelle known as lysosomes. How do we describe the shape of lysosome? Kindly check in your notes, the notes that I forwarded last time. Check if you can able to identify lysosome within the cell. They are spherical sac-like organelle. They are spherical sac-like organelles. Therefore, when we say they are spherical sac-like organelles, they are found in form that somehow they are spherical, but it look like a sac. It look like somehow a paper bag. Let's look at the function of lysosomes. The function of lysosome in a cell is to secrete lytic enzymes to destroy aged or unwanted organelles. Student, this is also a very, 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 very critical and also a very confusing organelle. Whenever you are told, you give the function. Don't just rush and start telling us that the function is to destroy worn out organelle. That's very wrong. Very, very, very wrong. It does not destroy. What's destroying this organelle? Unwanted organelle. Remember that we have just seen that a cell is a living component. It consists of living components. So there are some of the organelles that die when there is no more function, or if they age. There's a certain age that this organelle they might reach and they die. So you get that we have this very important type of an organelle known as lysosome, which play a very, very important role in that cell. Let's just imagine, let's say the mitochondria, the functions, it's already age to die, or it is just there. Let's say any other organelle like centriole, they've already played the role of cell division, and they age, they are just there. You get that this organelle will end up being removed because they end up accumulating in a cell. They become many. This is an example of human being. One day, one time, we will all die to give space for other generations. Therefore, this cell also needs to be emptied, at least to give the new organelles space to play the young one. Because let's say we have a mitochondria, it plays the role of energy production. It reach a time it become age, or it tired, or even can die. You see, if you can leave it there without removing or without exposing, they end up feeling or be many in that cell, and they are not playing any role. So this lysosome play a very, very important role in an organelle, are very important. So any organelles in that cell all have very importance. They all have very, very important role. They all have very, very important role. They all play a very important role. Therefore, you see that this organelle is very important because its work is to secrete lytic enzyme. This is an enzyme which help in destroying of this age or the worn out organelles. Therefore, it is also very important. Let's take an example. This question was in last time, I don't know, Form 3 exam, it's a very nice question. They were asked, give an organelles which are found in abundance in white blood cells. Students, you have asked yourself, what the function of white blood cells? What's the function of white blood cells? Before you rush, start writing, you're writing to me that centrioles, mitochondria. No, you have ask yourself, which organelle is found in abundant in white blood cell? You first ask yourself, what function of white blood cell? What's the function of white blood cell? So white blood cell play an important role of fighting diseases. Play an important role of fighting diseases. Therefore, if we play the role of fighting diseases or pathogen causing diseases in the body of living organisms, then which organelle is found in abundance in these white blood cells. Which organelle is found in abundant in the white blood cells? So the organelle which is found in abundant in a white blood cell is the lysosome. Why? Because the lysosome is the one playing the role of secreting lytic enzyme to destroy out worn out organelle, to destroy out age worn out organelle. So you get that it should be found in white blood cell in abundant, because you see now this white blood cell, its role is to fight pathogens causing diseases, or to fight microorganisms causing diseases. So you get that this lytic enzyme being secreted, it's the one encalving on the surface of this pathogen or microorganisms and to destroy them. So that's another part of application questions on lysosome. So you don't really expect to be asked, you give the function of lysosome. There must be an application questions on it. There must be an application question. Like for example, name organelles found in abundance in white blood cells. You can even be asked, you name organelle found in abundance. Maybe, for example, in muscles. You see, so you have to ask yourselves, muscles, for muscles to function, what does it require? Energy. So the one found in abundance is mitochondrion. Good. So now let's go to the next organelle. That's the ribosomes. Ribosomes, I think we have mentioned the ribosome when we are discussing about a rough endoplasmic reticulum. And we describe ribosomes as proribo, from the word ribo, a spherical shape, a spherical structure. So when we say ribosome, this is a ribo, it's a ribo, it's a structure like, and some are scattered within cytoplasm, while others are found on the surface of rough endoplasmic reticulum, as we have mentioned earlier. So apart from them found on the surface of rough endoplasmic reticulum, they are also scattered. They are also scattered on the cytoplasm. And student, I hope I had mentioned earlier the function of ribosomes. I had mentioned earlier the function of ribosomes. And I said that ribosome plays the role of manufacturing or synthesizing proteins. But when you are asked such kind of questions during an exam, you give the function of ribosome. You don't just rush and start writing synthesizing proteins or manufacturing proteins. It's very wrong. We say that site for protein synthesis, that is a biology KCSE marking technique. You see, in our books, it's also stated very well. In our textbook, it's also stated very well that they form site for protein synthesis. So we don't just say that ribosome are used for synthesizing proteins. No, very wrong. Remember that, students, there are some organelles whereby when you are asked, you give their function, we say site. We say site. We say site for this. But there are some, when you are told you give the function, you just go direct. For example, smooth endoplasmic reticulum, we say that its function is to synthesize and transport protein. We come to rough endoplasmic reticulum. I hope you are noting those important points. When we say rough, we say that its function is to transport proteins. But there are those which are very, very, very keen. You have to be keen, like mitochondria, ribosomes, chloroplasts. So when you are told you give the function of mitochondria, you say site for this. When you are told you give function of ribosome, you say site for protein synthesis. That's what we call KCSE marking technique. Therefore, ribosome do not synthesize protein, but it plays the role of doing what? Site for protein synthesis. For protein synthesis. Okay, students, can see we are doing on well. Let's move on. Golgi bodies. Golgi bodies are also referred to as Golgi apparatus. Golgi bodies are also referred to as Golgi apparatus. So, how do we describe, or how will we able to identify this organelle from others? Organelle from thousands and in that cell. So this Golgi bodies or Golgi apparatus are described as stack of membrane bonded tube-like sac. They are found close to the cell membrane. When you look at that, a generalized plants and animal cell, you will realize that these Golgi bodies or Golgi apparatus are always found near. They are always found near a cell membrane. They are not found on the central, neither on the middle part. They are found just next to the cell. So its work of this, we describe it as a stack of membrane. These are membrane which are stuck together, folded like, and then at the either side, we have some particles. We have some particles known as vesicles. So, have you ever seen ribs? Ribs. Or have you ever seen ribs? They are curve shaped. They are curve shaped. So that's a better way I can able to describe these Golgi bodies. So if you have ever seen ribs, kama hujaiona tafadhali enda buchari uoneshwe, enda usemi uoneshwe mbavu known as ribs. Uta realize that it is curve shape. Kindly, hiyo ndio kazi nimekupea. That's an assignment. Make sure tell your parents to buy meats parts enye atabuy ni ribs. Hiyo side ya ribs. Alafu kindly pick three bony part. Alafu you put them ikeleanisha pamoja, weke hivi, ikeleanisha ingine juu na weke ingine juu. I tell you, we have formed a structure of a Golgi body. We have formed a structure of a Golgi bodies. They are rib-like structure. So kindly, hiyo ni assignment nimekupea before we meet on next lesson on Tuesday. So make sure umechukua rib moja, weka chini, kaingine juu, weka ingine juu. Alafu hata unachukua thread, you tie the ends, and then you uweke hiyo as a study tool. You have formed a structure of a Golgi bodies. You have formed a structure of Golgi bodies. Okay, now students, let's look at function of the Golgi body in a cell. Let's look at the function of Golgi bodies in a cell. So, number one, this Golgi body has got several functions. Number one, I hope you are noting them. Number one, they play the role of package and transporting glycoproteins. Package and transport. They packed. First of all, they packed. After packing them, they can able to play the role of transporting. And then number two, they can also play a role of transport synthesized material out of a cell as secretion. Transport of synthesized material out of cells as secretion. And number three, they play the role of manufacturing lysosomes. I hope we have mentioned earlier lysosome. You know the function of lysosome and you know where it is applied. So they are formed by the Golgi apparatus or Golgi bodies. Good. Students, up to that point, we have done a lot. And before I move on to the next organelle, kindly Let me take you some of the important that you can note on during lysosome, ribosomes, and Golgi apparatus. Important notes. Note this. Note this. Ribosomes do not synthesize protein, but what does it do? What does it do? Form site for protein synthesis. So whenever you are told the function of ribosome, kindly avoid synthesize proteins. Avoid synthesize protein. It is very, very wrong. You should say site for protein synthesis. It's a reminder. Reminder on marking technique. Let's go to lysosomes. Lysosome does not destroy non-functional cells, but what does they do? Secrete lytic enzymes. Secrete lytic enzyme. And the lytic enzyme are the one playing that role of destroying this worn out organelle. Therefore, you don't say lysosome destroy non-functional or destroy worn out organelle. It is very wrong. You say that secrete lytic enzyme to destroy worn out organelles. Kindly, student, giving negative answers should be avoided. Don't give negative answers during examination, during answering your question, especially in biology. We don't accept negative answers. E.g., does not, is not, it lacks. No. We don't. We give alternative of that. And I say, and a good example I'm going to use is that of a cell wall and a cell membrane. We say that an animal cell, we don't say animal cell do not have a cell wall. When you give a comparison between a plant and animal cell, we say a plant cell have a cell wall and a membrane. But animal cell have a cell membrane only. Don't say a plant cell do not have don't say animal cell do not have a cell wall. It is wrong. You're supposed to say an animal cell, a plant cell have a cell wall and a cell membrane, while an animal cell have a cell membrane only. So that's a reminder of that. Good. Now, let's go to the next organelle. The next organelle that we are going to discuss is centrioles. Centrioles. How do we describe the structure of a centrioles? We describe the structure of a centrioles as rod-shaped structure located outside the nuclear membrane. Kindly check your notes. Go back to the structure of a plant cell or of a of an animal cell. Remember that last time I told you that centrioles, it's only found in animal cell, and chloroplast only found in plant cell. Note that those are very, very important points. Centriole, it's only found in animal cell, while chloroplast is only found in plant cell. So when I say centriole, you know that I'm only describing it on animal cells. And I say that they are located, outside the nuclear membrane. They form spidolites. I think we're going to learn this more in Form 3 topic on the topic of reproduction. We are going to learn more, and you are going to see what is this we call centriole. But now in Form 1 syllabus, I'm just going to mention the function. But in Form 3 syllabus, another topic on reproduction, you're going to learn more. You're going to learn more how these centrioles, how they look like, how do they play that role, you see. So for now in Form 1, you're only supposed to know that centriole plays the role of forming spindle fiber during cell division in animals. They form spindle fiber. In Form 3, you are going to see this spindle fiber. Get that during cell division, for this cell to divide, they form thread-like structures known as a spindle fiber. So what is forming that spindle fiber? It is these centrioles. They're centrioles. So you get that they are always, these centrioles in our cells, they're always occur in pairs. So for them to start forming spindle fiber, you get that since they appear in pairs, they start separating. One of the central go to the northern pole, and the other go to the southern pole. So they start forming what we call spindle fibers that help in separating. Maybe during, you're going to understand that in Form 3. For now, you can't understand. Whenever I try to explain, cannot understand because this one should be done practically. Okay, another function of the centriole, apart from forming spindle fiber during cell division, they also form cilia and flagella in our cells and organelles. Cilia and flagella. Students, what are cilia? Cilia, these are hair-like. Cilia are found on the trachea. They are also found of the nasal cavity. And the work of these cilia is to help trap dust. Let's say, for example, you live in a dirty environment, or you are just walking, all of a sudden you came across a dusty place. You see, these cilia, they're very, very important because they help in trapping these dust particles during sniffing in of this there. Therefore, these cilia are very important. So centriole play a role of forming cilia. And also cilia are also found in some organisms. They help in locomotion in some organisms. And we have flagella. A flagella. What is a flagella? It's known as a flagella This is a plurals, flagella, this is a plurals, and we have flagella, singular. So how will you differentiate cilia from flagellum? I say that cilia, these are hair-like, short. They are very short hair-like structures. But flagellum is a very long thread-like. Let's say an example of somebody with very long hairs, very long hairs, very long hairs. Like this mine, we have this very long flagellum. It's a hair-like structure, very long, a very long one, hair-like structures, and these are found mostly in organisms known as Eugelnum. We have an organism known as Eugelnum. This is also found in Form 3 topic. And you are going to learn more about Eugelnum in Form 3 topic, but now I'm just mentioning that this flagellum is a very long hair-like structure found in some organisms like Eugelnum, and the function is for locomotion. They are also used for defensive mechanism. They are also used in feeding. So you see, so those are function of Eugelnum. But then in Form 1, you only need to know function of centriole as the formation of cilia and flagellum in organism. And number 2, they form spindle fiber during cell division. Good. Now let's go to the next organelle, which is the chloroplast. Chloroplast is only found. The chloroplast is only found in plant cell. And chloroplast consists of green coloring matter known as chlorophyll. So student, if I mention now chlorophyll, you definitely know the function of the chloroplast. You'll definitely know the function of chloroplast. So, first of all, I remember when in the beginning of the lesson, when I was describing the mitochondria, we had also I had also give you a tip idea about the chloroplast. So we say that chloroplast, we also describe a chloroplast as an oval shape surrounded by 2 membranes. But we say that the membrane of this chloroplast, they are smooth. They have smooth membrane. They have smooth membrane. It's not like mitochondria. We say that the inner membrane of mitochondria, it is highly folded. But when you look at this chloroplast, we get that the 2 membranes are smooth. They are smooth, and therefore we have said that they contain chlorophyll, which is a green coloring matter that is used for photosynthesis. They contain chlorophyll, a green coloring matter that is used for photosynthesis. And now, if you are asked, give the function of chloroplast, then how will you give the function? How will you give the function of it? You give the function as site for photosynthesis. You don't just rush and start saying they contain chlorophyll that trap light energy that's used for photosynthesis. No. If you are told you measure or you give the function of chloroplast, you say that site for photosynthesis. Simple as that. You score your mark. As simple as that, you score your mark. So the next organelle that we are now going to discuss is the vacuole. The next organelle, it's the vacuole. And last time, I was surprised that some students were telling me the vacuole is only found in plant cells. No, vacuoles are found in both plants and animal cells. The only difference is that in plant cell, the vacuole is centrally placed. It is large, visible, very large. You can able to see and you can able to identify because it is large and centrally placed. Large and centrally placed. But when you come to animal cell, you get that these vacuoles are very small. They are very small and they are found on the peripherals. These are ends. These are ends of a cell. So when you look at the vacuoles in animal cell, they are very, very small. They are small, and apart from them being small, they might be permanent or they might be permanent or temporary. They disappear. In some cases, you can get that they are not there. But I'm not meaning that vacuoles are not found in animal. They are found, they are there, they are small, but they're not permanent. They're just temporary, but they are there. They are there, they disappear. But in animal cell, it is there and is permanent, very large, a very large central structure and permanent. And it is centrally placed. So we describe vacuoles as sacs that fill with fluid. They always have a fluid medium known as sap. S-A-P, known as sap. Hence, sap vacuole. Sap vacuole. So, within a vacuole, we have a fluid-like medium known as sap vacuole. And a membrane that is surrounding this vacuole is known as tonoplast. You know, I was surprised that some of you were telling me last time that a tonoplast is an organelle. It's an organelle which is not found in animal. No. A tonoplast is a membrane. You see, we have what we call a cell membrane surrounding the whole of the cell. Cell of a plant and that of animal. So we also have here what we call a tonoplast. A tonoplast is also a membrane surrounding a vacuole. And within that vacuole, we have a medium-like fluid known as sap vacuole. Sap vacuole. And this sap vacuole is very, very important because it contains a solution of salts and sugar that contribute to osmotic pressure of a cell. So, this is very important. You get that we have like organism, like unicellular organisms. We have what we call food vacuoles, and we have what we call contractile vacuoles. So we have two types of vacuoles in unicellular organisms. We have what we call contractile vacuole. We have contractile vacuole, and we have and we have food vacuole. So kindly, student, listen carefully and note this. Give me an example of a unicellular organism. A good example of a unicellular organism that most of you know, it's amoeba. Is amoeba a plant or an animal? Automatically, this is an animal. So you see, so vacuole is found in both plants and animal cells. It's found in both plants and animal cells. And we say that within this vacuole, we have a fluid medium known as sap. A sap is a solution of salts and sugars that contribute to osmotic pressure of this organism. So, and we have two types of vacuoles in these unicellular organisms like amoeba. We have what we call food vacuoles, and we have what we call contractile vacuole. So, whenever you are told you give function of vacuole, don't just rush and start saying that it contain a fluid medium. No. How will you answer this kind of a question? The first function of a vacuole, we say that you have to mention, because we have just said that there are two types of vacuoles. We have food vacuoles and we have contractile vacuole. When you are told you give function of vacuole, you say number one, food vacuoles. Food vacuoles stored and digest food in unicellular organism. Store and digest food in unicellular organism. Number two, we have what we call contractile vacuole. What's the role of contractile vacuole? Its role is to excrete excess water and mineral salts from the organism. Let's take an example of amoeba. Amoeba is an organism that live in water, living inside the water. You see, this organism experience a very, very big problem. That's the habitat of this organism, but in real sense, it is really suffering because it is keeping on absorbing excess water in its body. So how will this amoeba able to manage its problem? How will it able to cope with its problem? So they develop what we call contractile vacuoles. So these contractile vacuoles in this amoeba are very, very important, because after this amoeba absorbing excess water, they cannot stay with this excess water in its body because they swell. Therefore, they come to the water surface and this contractile burst. When it bursts, it releases this water. So it renews again this contractile vacuole. So that's how the amoeba cope up with the problem of absorbing excess water. So after it absorbs this water, it comes to the water surface, and the contractile vacuole bursts to release this water. So vacuoles in a cell of a living organism is very, very important. Very, very important. Good. Now let's go to our last organelle. That is the nucleus. This is our last organelle in this topic. Organelle nucleus. Sorry, this is our last organelle that we are going to discuss. Nucleus. Nucleus is found in both plants and animals. The only difference is that in animal cell, the nucleus is centrally placed. It is centrally placed. But in plant cell, it's peripheral. It's at the side. It's on the peripheral. It's on the. It's just next to the cell membrane, in between the cell membrane and the vacuole. That's where you can get the nucleus. So under electron microscope, the nucleus is seen to have a double membrane called a nuclear membrane. When you look clearly, when you look clearly, when you look clearly, on the microscope, you can able to see a nucleus, and the nucleus, it is seen to have a structure surrounding it, and this structure seems to have some gaps in between. So meaning that's a structure of a nucleus. It is surrounded by a nuclear membrane. Those gaps, they are not as nuclear pores, and very important because they play the role of allowing movement of substances in and out of a cell. And apart from having a nuclear membrane, nuclear pores, we also have a patch particles, a dot. It's not, how can I describe it? A particles on the, not at the central, but on the top of a nuclear, known as nucleolus. So students, don't confuse between a nucleus and nucleolus. So we say that the membrane has pores which allow the passage of material in and out of the nucleus. The nucleus is made of viscous fluid known as nucleoplasm, in which the nucleus and chromatin are suspended. So when you are told you give the function of the nucleus, the main function of it is to control all activity of a cell. And by now you know what are the activities of a cell: energy in production, the store of out organelle, synthesize the role of photosynthesize proteins, synthesize fats and lipids. Therefore, we say that the main function of nucleus is to control all function of a cell, to control all the function of a cell. So that's all, and I believe you have understood and you have enjoyed the lesson. Now we have finished discussing the living component in a cell. Kindly note and remind yourself again and again so that they remain in your fingertip. Whenever you are asked any question based on organelle, don't fail me. You can able to give us perfect answers. So during our next lesson, we are going to discuss differences between plants and animal cells. So kindly question. Attempt the following question. Question number 1. State at least 5 activities of the cell that are controlled by the nucleus. 5 marks. State at least 5 activities of the cell. State at least 5 activities of the cell that are controlled by the nucleus. 5 marks. Question number 2. List the organelle found in both plants and animal cells. Number 2. List the organelles found in both plants and animal cells. List the organelle found in both plants and animal cells. So kindly attempt those questions. And before we continue from where we are going to continue next week, the first thing we are going to do is marking those 2 questions. Bye. And I hope you have enjoyed the lesson. Thank you.